My name is Hajra Jamal, and I'm really thankful to be here today. When was the last time you had a bowl of rice? From paella to biryani, rice is an important part of our sustenance and culture. 3.5 billion people around the world depend on rice as an essential source of nutrition. In Asia, where 600 million people live in extreme poverty, rice consumption exceeds 200 pounds per capita annually. In many Asian countries, people eat a bowl of rice with every meal. Rice also plays a social role, such as in Hindu wedding ceremonies and in the sacrificial rituals of the indigenous Ifugao people of the Philippines. The Ifugao people, in particular, center their livelihood around growing rice. Their villages are situated in rice terraces, such as in Banu. For the Ifugao people, rice is an important part of their sustenance, economic welfare, and it serves as an offering to the Bulul, the Ifugao rice god. Thus, environmental conditions that compromise the agricultural prosperity of rice also compromise the social and cultural practices of those who depend on rice. As atmospheric carbon dioxide rises and the polar ice caps melt, our environment suffers in many ways. Climate change not only threatens the environment, but the prosperity of humanity itself. As the global population grows and the integrity of our ecological resources decline, we are faced with the challenge of tackling global food security. Scientists estimate that by 2050, the global population will rise from 7.5 billion to 10 to 11 billion. These complex dilemmas present a global economic existential crisis. How do we continue to meet the increasing food needs of a growing population in a sustainable and equitable manner? How do we make sure that there is rice on the table for everyone who depends on it? Part of the answer lies in closing the yield gap. There is a disparity between the ideal crop yields that should be obtained with current technologies and management and the realized productivity of agriculture. Second, we must develop crops that are resilient enough to withstand climate change. Environmental stressors such as drought, flooding, and high temperatures can be addressed through transgenic approaches. Transgenic approaches would utilize staple crops such as rice. Rice can be bred with the foreign DNA of other plant species so that rice has the genetic means to withstand specific environmental challenges. Scuba rice is the poster child for transgenics in agriculture. Targeting South Asian regions prone to flooding, the International Rice Research Institute bred several varieties of rice to introduce a submergence-tolerant gene, SUB1. For years, farmers in India and Bangladesh have been growing this transgenic SUB1 line variety of rice to maintain their yields during floods. And lastly, the global agricultural system must remedy its environmentally harmful practice of using excess industrial nitrogen fertilizer. The nitrogen in fertilizer, as well as water, are two key elements of plant growth and agricultural prosperity. Issues involving water, such as floods and drought, are clear consequences of climate change, but the role of nitrogen is more subtle. Unlike other nutrients that leak into soils from rocks, the source of nitrogen comes from the atmosphere. Plants naturally rely on nitrogen-fixing and nitrifying bacteria among their roots to convert the nitrogen gas into forms that are accessible to plants, nitrate and ammonia. Current agricultural systems use industrial fertilizer to apply ammonia and nitrate to soils that have been stripped by repetitive cultivation. The Haber industrial process for artificially fixing nitrogen gas into ammonia is environmentally taxing in three ways. It leads to greenhouse gas emissions, consumes 2% of non-renewable energy annually, and leads to eutrophication. Eutrophication occurs when crops are not able to use all of the nitrogen in fertilizer and it leaks into bodies of water, causing algal blooms. These algal blooms deplete the water of oxygen, thereby causing anoxic dead zones, harming aquatic life and making that water unpotable. Thus, by closing the yield gap, making more resilient crops, and reducing dependence on industrial fertilizer, the global agricultural system can work towards addressing food insecurity in a sustainable manner. As seen with the example of scuba rice, transgenic approaches are widely successful. For the past year, I have been working in the NYU Cruzy Lab on a similar endeavor. We have been using a transgenic approach to tackle food insecurity, recognizing that crops need to be improved to withstand climate change and the need to reduce dependence on fertilizer. Our research gets to the root of the problem by focusing on the two key elements of plant growth, nitrogen and water. Our research aims to improve the ways in which plants use and take up nitrogen and water to help them grow in unideal conditions and adapt to a rapidly changing climate. Our lab uses this weed, Arabidopsis thaliana, a model plant organism, basically the lab rat of the plant research world. I remember the first time I started in the lab, I was told that the Arabidopsis seeds are the size of a 12-point font times New Roman period. I was also asked if I played an instrument, because sowing the seeds with the pipette required a lot of dexterity. I thought they were just trying to scare me, but I was so wrong. It took me months to figure out how to sow those seeds properly, to figure out how much water and how many seeds to suck up into the tip of the pipette, and how much pressure to maintain at the top. Every time we run an experiment, we sow about 200 seeds. And the first time I did that all on my own, my mentor couldn't believe it, and it felt so good. Arabidopsis is useful for several reasons. It has a quicker life cycle than rice, allowing us to run more experiments and make discoveries faster. Its genome is also fully sequenced, and compared to rice, mutations of its genes present clear phenotypic characteristics, which are readily apparent effects at the physiological level. Because Arabidopsis and rice share homologous genes, which are ones that are evolutionarily conserved, we can translate our findings of the nitrogen and water interaction in Arabidopsis to rice. The nitrogen and water interaction demonstrates how plants are able to modulate these two factors to facilitate their growth. Through 4 experimental conditions with either lower high nitrogen and lower high water, we found that high nitrogen is actually detrimental to growth when water levels are low. This is a significant piece of information for farmers experiencing dry seasons. There are a number of ways by which plants use, take up, and transport nitrogen. Part of our research focuses on a specific gene that encodes a particularly limiting nitrate transporter that also has sensing ability, which is depicted at the top. One mutant of this gene does not have sensing nor transport ability, and yet another mutant has sensing ability but not transport. And to our surprise, the mutant with doubly compromised function is actually drought resistant. The canonical drought survival experiment best illustrates this finding. So after growing the plants in normal conditions, we then starve them. And after rewatering the plants, we see that the doubly compromised mutant is the one that essentially comes back to life and has the best rates of survival. This not only demonstrates the nitrogen and water interaction, but shows that this doubly compromised mutant is able to survive in drought conditions because it is able to limit its nitrate intake. These research findings present 3 points of benefit to farmers and their crop yields. First is the piece of information that during dry seasons, it is best not to use fertilizer or excess fertilizer. Secondly, the CHO 1.5 mutant, which is the one with doubly compromised functions of transport and sensing, can serve as a potential candidate for transgenic approaches, similar to scuba rice. And lastly, the genes and mechanisms by which plants use and take up nitrogen and water can be used as biomarkers. Similar to how cancer biomarkers predict risk, these biomarkers would allow farmers to predict low yields. By bringing the tools and the knowledge of the laboratory to the field, our research helps address food insecurity, grants agency to local farmers, and helps keep the religious and social practices of those who depend on rice intact. Thank you.